Okay, so let's continue on. We have done 2 of the 4 phases of a muscle contraction cycle. We did the excitation and then we did the excitation coupling phase. Now we're in the 3rd phase of contraction, which is the part we've previously talked about. So where we left off was with an energized myosin head because it already had ATP bound to it. And now we have calcium that's released and the cytosol. Okay, so that calcium had bound to the troponin to pull the tropomyosin, exposing the actin binding site. So the myosin head, which is energized, forms that cross bridge and does the power stroke, where the myosin head is bending, pulling the actin towards the M line, towards the center of the sarcomere. And so that sarcomere is contracting because it's pulling on both directions, both ends, so it's contracting just a little bit. Once it's contracted, the ADP and the inorganic phosphate on the myosin head are released. The myosin head is still stuck in place. Meanwhile, the calcium ions have removed themselves from the troponin. They're back in the cytosol. The myosin is still stuck to the actin, okay? So what has to happen is a new ATP has to bind to the myosin head. That causes a destabilization of the cross bridge. The myosin head gets recocked, okay? Because that ATP was hydrolyzed by the ATPase in the myosin head. Now, what the picture shows is that calcium is being reabsorbed here on the left side into the sarcoplasmic reticulum, while calcium is being released from the right side. Because remember, the calcium release is linked to the voltage-gated sarcolemma receptors. So this process will continue over and over again as long as we continue to have an action potential in that muscle cell, and as long as we continue to have calcium being released to go to bind to the troponin, and 3rd, as long as we keep on having ATP to rebind to myosin heads so it can be cocked and ready for its power stroke. So yes, the sarcoplasmic reticulum can be both releasing calcium in one area, the terminal cistern, and reabsorbing it along the rest of the sarcoplasmic reticulum at the exact same time. So when myosin is released from the actin, that's only happening to that one myosin head. All the myosin heads are not being released simultaneously because each one has its own timing as to when it formed the cross bridge. Okay. And so if all the myosin heads would have their cycle, their power stroke and their recovery stroke, all at the exact same time, then the muscle fiber would shorten 1%. So I hope by now you've realized we have to have a lot of these ratchetings going on. Because many cycles, 40 cycles for all of them would be required for a fiber to shorten 40% of its length. Okay, and going back to how fast this happened, each myosin head can do the, go in, do my power stroke, do my power stroke, have a new ATP bind, do my recovery stroke, ready again, power stroke, new ATP bind, recovery stroke. It can do that 5 times per second. So now that we've gone through all this detail, I want you to look at this picture for a prolonged period of time and make sure you're talking about actin, tropomyosin, troponin, calcium, myosin head, ATP, ATPase, ADP, and PI, or in other words, inorganic phosphate, and you should be able to see everything that's going on at this point in time. So, muscle fiber relaxation occurs when there's no more action potential, okay? If there's no action potential coming off of the neuron at the neuromuscular junction, then there's no acetylcholine being released, and acetylcholinesterase is going to destroy whatever was in the synaptic cleft, which means there's no acetylcholine to bind to those cholinergic receptors, which means those ligand-gated channels will not be opening. It also means there's no impulse traveling down the sarcolemma to release any new calcium from the sarcoplasmic reticulum, which means the sarcoplasmic reticulum can only reabsorb the calcium. The cytosolic calcium levels are going to plummet, so there's no calcium available to bind to troponin, which means troponin is just staying, which means troponin has moved back so that tropomyosin is covering the active binding sites. Meanwhile, there's no muscle tension because the myosin heads are cocked and there's no place for them to go. So the muscle fiber no longer has tension and it is relaxed. Whew. So there's a lot of negatives involved for muscle fiber relaxation. So now here is a slide that's putting all 4 phases together. Starting up where number 1 is, that's where the terminal bouton is, okay? And so kind of talk yourself through the entire process, freeze it here. If you get stuck, go back and look at all the steps. Look at all the pictures that we've talked about for all 4 phases, okay? And I will meet with you shortly. With this, we're done with the neuromuscular junction and the sliding filament theory, and we're going to back up and talk about something a little lighter for a while. This is the end of the heavy phases stuff for today. Thank you for all your hard work, and I will see you shortly.